Hello again. Let's continue with our discussion of this process of respiration, and what we want to focus on now is an event called chemiosmosis. And we're going to look at a particular kind of chemiosmosis. It's what also could be referred to as oxidative phosphorylation. All right, now you have this illustration as part of your handout, so make sure you find this. This will be important for you to have handy while we're talking our way through this, okay? So go ahead and get that. Pause me for a moment, and let's get ready to talk about this important energy-producing event. Very interesting event also. First of all, we're going to have to talk about our electron carrier molecules again. Remember what we said. We have these modified dinucleotides, NAD, NADP, FAD. All three of these are modified dinucleotides. And we're going to see that two of them are going to get involved in this process of chemiosmosis we're going to discuss. It, in your cells, uses just two of these. If we added bacteria into the mix, then we could actually talk about variations in which all three of these electron carriers are being used. Now, what do each of these electron carriers do? They carry a package of material, cargo. They're carrying a pair of electrons and a pair of hydrogen ions. Now, you could also think of this, remember, as two hydrogen atoms, but we're going to be thinking about it in terms of these two components, because the electrons are going to move one way through this process and the hydrogen ions are going to move a different direction. All right, so let's see where these things are going to be going. These electron carriers are going to arrive at the electron transport chain in your cells, in eukaryotic cells. Of course, this is in the inner membrane of the mitochondria. Okay, if this were a prokaryotic cell, remember they don't have mitochondria, so this would have to happen in their plasma membrane, their cell membrane. So these electron carriers arrive in their reduced form. They're carrying their loads of material. When they arrive at the electron transport chain, they're going to deliver their package, their cargo, to the first molecule of the electron transport chain. Remember what they're dropping off? 2 electrons. That's the key thing that are being passed to the electron transport chain. Simultaneously, they let go of the 2 hydrogen ions that they're carrying as well. As a result, they move to their oxidized state, and at this point, all these carriers could float back, diffuse back through the fluid of the cell to a location where they could become reduced again. So remember, they're being reused. They don't do this just once. They pick up materials at one location in the cell. Do you remember where those NADs became reduced? You might want to go back and review that, oh, that's right, we made some NADHs during glycolysis. We made some NADHs during the acetyl formation step, and we made some during the Krebs cycle. Those are those locations where the oxidized forms of these molecules would float back to be reduced again and repeat the process. So make sure you think about this as a cyclical event. These NADs being used and reused over and over again by a cell. Well, now let's move on and see what's going to happen to these electrons. As I mentioned, they get passed to molecules of the electron transport chain. You'll also see this referred to as the electron transport system. Now, it requires that there's some kind of membrane involved. Now, if we're talking about a mitochondrion, then we're talking about this electron transport chain being embedded in the inner membrane of the mitochondrion. What do we have? Well, they're integral transmembrane proteins primarily. And we're going to be most interested in the components of the electron transport chain that are going to be proton pumps. They're active transport, right? Active transport transmembrane proteins. So we're going to be seeing some kind of process of pumping going on. As a matter of fact, what we're going to say is that these molecules have the ability to use energy provided by electrons to push or pump hydrogen ions across the membrane that they're embedded in. So that's the way you want to think about what's going on, that these proteins are pumping hydrogen ions across a membrane. This is one of those good examples of a cell creating a disequilibrium condition. Remember what disequilibrium means. More of something on one side than the other side of a membrane is the way you could think of it here. We're not talking about things flowing equally back and forth across a membrane. Instead, we're pushing them across the membrane one way, and then you'll see that we're going to let them flow back across the membrane in the other direction, releasing the energy that they've gained by being pumped across the membrane. Right, now here's one way we can represent this electron transport chain. It's a very simplified representation. And notice what I've tried to show is that we could think of at least 3 locations within this electron transport chain where there are these proton pumps, these transport proteins that are going to push hydrogen ions. Notice hydrogen ions from a low concentration area to a high concentration area. To do that, remember, if I'm going to move something against its concentration gradient, it's going to require additional added energy, energy above just the kinetic energy that's found in these hydrogen ions. Where does that energy come from? Well, it comes from the pair of electrons that are being passed down the chain while the hydrogen ions are being moved across the chain. So make sure you get those 2 directional movements clear in your mind. Now imagine, if you will, if I was one of these protein transport proton pumps. All right, what do I do? Well, I'm sitting there in a particular shape or a particular conformation. And up against me, let's say that at a pocket on the side of me, NADH comes along and gives me 2 electrons. Well, think back to what we said is the determinant of the structure of molecules. Isn't it the position, the distribution of the electrons they have? Where are my electrons? Remember, all my covalent bonds are just pairs of shared electrons. And now what's happened? This molecule, NADH, has come along and given me 2 more electrons. That's going to change my shape. Think about this sequence of events. I receive 2 electrons from NADH. I change shape so that I grab a hold of 2 hydrogen ions from the solution floating around, and I change shape. By changing shape, think about what I've done. I've started with hydrogen ions that are at this location on one side of a membrane. By changing shape as a result of the energy of the electrons, I pull those hydrogen ions across the membrane. I'm pumping them from a low concentration region, like the matrix of a mitochondria, to a high concentration region, the intermembrane space of the mitochondria. Make sure you think about these locations when you look at the pictures in your book of a mitochondrion. So, it's the power from these electrons causing me, as this transmembrane protein, to change shape that pulls the hydrogen ions across the membrane. Now, I'm just one of these transport proteins, and adjacent to me would be additional transport proteins. So what do I do with the 2 electrons that I've just received from NADH? I pass them to the next molecule. It goes through a shape change. It pulls hydrogen ions across the membrane as well. Those electrons are passed to a 3rd proton pump. Again, pumping occurs. Now, we don't understand exactly how many hydrogen ions get pushed across the membrane for each pair of electrons that gets passed down the chain. But we do know that it's enough power. In other words, I've created enough of a hydrogen ion gradient that I will eventually be able to produce 3 ATPs. So you want to keep that in mind, and we'll talk about it again. Let me recap what I'm just saying. That for every 2 electrons that are delivered to the chain, enough hydrogen ions are pushed across the membrane to eventually provide enough power to make 3 ATPs. And we're going to use that conversion factor later on. Well, what happens to the electrons then? Notice they come out of the end of this chain. They have to go somewhere. And so what we find is that at the end of this electron transport chain, there's going to be a molecule called a final electron acceptor, an atom or a molecule that's going to capture those electrons as they come squirting out the end of the chain. Now, in organisms like ourselves, we use oxygen. Matter of fact, we do a type of oxidative phosphorlation where oxygen is our final electron acceptor, and therefore we would be classified as doing aerobic respiration. A little bit more about that in just a moment. So, let's recap this very quickly. I have electrons moving down the chain. I have hydrogen ions being pumped across the chain. The electrons start with NADH or FADH2, or in bacteria, Some use NADPH. The electrons eventually end up being added to a final electron acceptor. So we're going to want to think about that as we move on to other parts of this process. Now, these final electron acceptors, as I mentioned, we use oxygen. As a result, we are said to do aerobic respiration. And what we're going to find is that when we look for the total production of ATPs from aerobic respiration, we're going to get somewhere around 36 to a maximum of 38 ATPs for every 1 glucose that's completely broken down by aerobic respiration. And we'll do a summary table where we'll take a look at this, and we'll see where those different ATPs have come from. Remember already, by the time we got here, we had made 4 ATPs, 2 in glycolysis and 2 in the Krebs cycle. So we're looking for an additional production of about 34 more ATPs as we complete this story. Here's the problem. Here's the drawback of using oxygen. Whenever I use oxygen as a final electron acceptor, I generate a large amount of toxic waste material. It's what people refer to in a general sense as oxidants. As a matter of fact, that's why people recommend that you take antioxidants. It's one of the things that it benefits is helping to clean up this toxic waste generated every time an organism uses oxygen as a final electron acceptor. One example of this toxic waste is hydrogen peroxide. Now, hydrogen peroxide is a potent poison. It's a very strong oxidizing agent. It wants to steal electrons from other molecules. I can't have this hanging around in my cells. It will cause the deterioration of my molecules. So I need to clean it up. And you probably remember from what you dealt with in lab that you have a special enzyme for cleaning up hydrogen peroxide. It's called catalase. So notice what I'm suggesting. There's benefits of using oxygen as a final electron acceptor, but there's also drawbacks. And what are the drawbacks? Toxic waste. I have to clean it up somehow. Now, if I use a different final electron acceptor, some other kind of inorganic substance other than oxygen, let's say something like nitrogen or sulfur, they're the 2 most common ones, then I'm going to be doing a process called anaerobic respiration. Very similar to aerobic respiration, also has chemiosmosis in it. The key difference is the final electron acceptor. What we find is that those organisms that do anaerobic respiration are always going to be getting less than 36 ATPs for their glucose. And also we find this process of anaerobic respiration exclusively in bacteria, prokaryotes. Excuse me, I should also include the archaea. So we don't see anaerobic respiration occurring in eukaryotic organisms. We can't do that in our cells. All right, so now let's consider what happens to those hydrogen ions that I've pushed across the membrane. This is a simplified representation trying to represent a portion of that total illustration that you are looking at right now. Notice here I'm just emphasizing the enzyme system at the end of this process of chemiosmosis. This molecule right here that I've drawn as a light bulb kind of shape is ATP synthase. ATP synthase, as its name implies, can synthesize ATPs. It can take ADP, force a phosphate group to attach to it, and make ATP. But that requires energy. Remember what we said about that ADP and phosphate group? They don't want to go together. I'm going to have to push on them very vigorously to force them together. It takes a lot of energy to make that bond between ADP and the phosphate group to produce the ATP. What's going to provide that energy? The key is the movement of these hydrogen ions. Remember what we did? We created a hydrogen ion gradient. You could think of it as being analogous to a dam across a river. Think about what we do. We put a dam across a river and we create a concentration gradient, don't we? Lots of water on one side, less water on the other side. We open a hole in that membrane and the water comes rushing through. Its potential energy gets converted into kinetic energy, and we use that to make electricity. Well, the cell's doing a similar kind of thing. This transport protein enzyme synthesizing complex that we call ATP synthase allows the hydrogen ions to flow through the protein, and as they do, the protein rotates. Isn't that amazing? As it rotates, you could visualize that it has an active site that has 2 parts to it. One part fits ADP, one part fits a phosphate group, and as the protein is caused to rotate, it squeezes those two molecules together. It helps add energy to those substrates so that the bond can be formed, so that we can make that ATP molecule. It's the rotation of the ATP synthase as it turns that forces together the ADP and the phosphate group. And it's the flow of the hydrogen ions that rotates the protein. The protons, the hydrogen ions moving, we actually refer to as a proton motive force. We're saying those protons are moving with such force that they can rotate that enzyme system. That's how we make the ATPs. Here's the interesting relationship. Every time an NADH drops off its 2 electrons to the electron transport chain, that provides enough power to push enough hydrogens across the membrane so that I can cause that ATP synthase to make one full turn. In one full turn, it produces 3 ATPs. So we would say that for every 1 NADH that gets to the electron transport chain, enough power is released to make 3 ATPs. Now, FADH also does a similar thing. It drops off its electrons at the electron transport chain. The problem is this: it drops off its electrons to the second proton pump. Not as many hydrogens get pushed across the membrane. As a result, not as many ATPs get made. But we're going to use the conversion factor of saying that for every 1 FADH that gets to the electron transport chain, it releases enough energy to be able to make 2 ATPs. So we're going to use that conversion factor later as we talk about the total production of ATPs from that one original glucose molecule we had. Remember, we had 10 NADHs. That should be equivalent to what? 30 ATPs, right? 10 times 3. We had 2 FADHs, and we said that each of those provide enough power for 2 ATPs. So for those 2 FADHs, we should get 4 ATPs. Notice there's our 34 ATPs we were looking for before. That's our final payoff from the energy that we stripped from the glucose molecule. So what are we saying? Chemiosmosis, this process, is going to power most of the ATP production in the cell. We're saying that electrons from electron carriers power the electron transport chain and move to a final electron acceptor. That powers proton pumps that move hydrogens across a membrane. The hydrogen ions diffuse back across that membrane through ATP synthase, cause its rotation. That provides the power to make some ATPs. Okay? So notice that this was chemiosmosis, and it began with the oxidation of NADH or FADH. So we can also call this oxidative phosphorylation. We made ATPs using energy from oxidizing an electron carrier molecule. Here's an overview. Take a look at it for a moment and see if it makes sense. This is a little bit different than our representation, but it's more similar to the one that you have in your book. And our starting point, NADH being oxidized, electrons moving down the chain to a final electron acceptor, hydrogen ions, protons being pushed across the chain, the hydrogen ions creating a proton motive force that flows back through ATP synthase, and we generate ATPs. It's a beautiful system. Living things on this planet are dependent upon different variations of this chemiosmotic process. And all we've been looking at is just the example of what happens in your mitochondria. Okay? Aerobic respiration. If you want to review this and look at the whole process that we've been talking about, let's just go back and recap our ATPs. Remember, 2 from here, substrate level phosphorylation. 2 from here, also substrate level phosphorylation. And then finally, oxidative phosphorylation giving us 34. Notice what our total is. The maximum ATPs that can be produced, 38 ATPs. All right, so we've completed the process of respiration. And I'd just like you to consider one other sort of little side idea about this. Remember we talked before about venoms and poisons and toxins, and we talked about how commonly they interfere with enzyme activity. Well, here's an interesting example of how some molecules we consider to be poisons interact with the electron transport chain. There are some, like rotenone, which is a potent poison, especially of organisms that are in aquatic environments, and you can see what it does. It blocks one of the steps of electron transport. If I can't transport electrons, my ATP production is shut down and I'm in trouble. It kills me. Something like carbon monoxide or cyanide, they bind to one of the last few molecules of the electron transport chain, and the electrons can't get past to the final electron acceptor. The result is still that my electron transport chain is all backed up. I can't make ATPs. I die. Some of these poisons act very, very rapidly. If you're familiar with the effect of cyanide on someone, you know that they can die within minutes of being exposed to that poison. So here's two interesting examples of poisons that affect, again, proteins, but not enzymes this time, instead members of the electron transport chain. All right, so think about what we've looked at. A structure, a group of molecules working together. Electron transport chain, ATP synthase. Electron carriers had to deliver high-energy electrons to this system to provide power to it. What was the power used for? To pump protons across a membrane. So we say that oxidation of the electron carriers provided the power for this process. That's why it's called oxidative phosphorylation. We also said that we could think of the general event, creating a hydrogen ion gradient with an electron transport chain, and then using that to power ATP synthase, the production of ATPs, as a chemiosmotic process. Also remember that we said one key distinction between the different forms of this process was the type of final electron acceptor. Oxygen in aerobic respiration, some other inorganic substance in anaerobic respiration, most commonly something like what? Nitrogen or sulfur. Very important process. You're going to want to review it. Practice trying to draw the entire process for yourself. See if you can recreate some of these illustrations from memory. It'll be good practice for the exam. Thanks very much. Look forward to talking to you next time.